Thank you very much. I'm really glad to be here and to be able to present some of the work that I'm doing and to be able to, more importantly, be able to help you guys understand and learn a little bit about molecular modeling, not just in terms of glycosaminoglycans, but a little bit in general, although this talk will, of course, be focused on GAGs. So what I'd like to do is to be able to go through and talk a little bit about the structure of GAGs first, and then I will explain some basic fundamental principles that we use in molecular modeling, and then be able to relate that to the actual modeling of the protein-GAG interactions and how we study them. Okay. All right, well, Dr. Desai told you a lot about the biological roles and the importance of GAGs in the last talk. I just had this slide here to show you just the phenomenal diversity of not just the structure, but of the interactions that take place. And this is just a very small sample of a couple of different important GAGs. Heparan and heparan sulfate and dermatan sulfate can interact with many different protein partners to affect many different biological responses. Perhaps one of the best known is anticoagulation, where it interacts with antithrombin and heparan cofactor 2, with annexin, also in the process of anticoagulation, where it competes with receptors on platelets. Umesh mentioned the C1 inhibitor in the last talk, complement activation. There's interactions with glycoproteins that are associated with viruses, glycoproteins B, C, and D of the herpes simplex virus 1, and also the very well-known GP120 entry protein that's part of HIV. Also, there's important interactions with cytokines. Platelet factor 4 and interleukin 8 are two that we're really interested in, and that one of our other principal investigators is actively researching. And also growth factors, cell proliferation, etc. These would be fibroblast growth factors, vascular endothelial growth factors, and the like. Okay, so just a little bit of background on glycosaminoglycans, affectionately known as GAGs. By the way, I thought our PEG, and I'm not alone in this, I thought our PEG should have been called the GAG PEG. But it just kind of rolls off the tongue a little bit more easily, don't you think? But HSPG-PEG is good too. Nice idea, Prob. So glycosaminoglycans are, of course, linear, that is, unbranched chains consisting of repeating disaccharide units. And these are variously substituted with acetyl groups and sulfate groups and can have different ring conformations. They can exist free or in solution or covalently linked to proteins and proteoglycans, in which case they're known as proteoglycans. They're phenomenally diverse with regard to the saccharide ring conformation and substitution pattern. And even within a single GAG, the substitution patterns are not uniform. There's regions that are more highly sulfated, regions that are not so sulfated, regions that are acetylated, and the like. And unlike other biopolymers, there's no genetic code that determines the precise GAG, I should say, conformation or substitution pattern, although there are several enzymes that are involved in the post-modification of GAGs. Okay, so what I'd like to do now is just take a step back and just give everyone kind of a refresher on the structure of GAGs, because this will be central to the understanding of to their modeling and their interaction with the proteins. So let's start with monosaccharides. And I'll focus on the aldoses. So for the aldoses, we have an aldehyde, shown here, reacting with an alcohol to give a hemiacetal. Hemiacetal is just a carbon with 4 substituents. There's a hydrogen, an R group, which is carbon and then something else. And then there's an OH group and an OR prime group, which is another carbon, something else. Okay, so this is hemiacetal. If this would be Anybody know what this would be if this was OR double prime? Acetal, just a regular acetal. This is hemiacetal, hemi meaning half. So one of the oxygens is substituted as something, the other just has hydrogen. Okay, so these two can react to give the hemiacetal. And this reaction is completely reversible. So it can also disassociate back into its original aldehyde and alcohol components. Well, this little figure shows that these two things might be different molecules, but it doesn't have to be that way. The alcohol and the aldehyde can be attached to the same molecule. Let's, for example, take this molecule. Let's put a 6-carbon chain here, and we'll put the aldehyde on one end, and we'll put the alcohol down here. Well, they can also interact in an intramolecular fashion, and this is also reversible. And then finally, what let's do is let's decorate these other intervening carbons with other hydroxyl groups. And so what I've shown here is this particular one is glucose. And that can be, this is what's called a Fischer projection. And I don't know if anybody remembers how the Fischer projection works, is that basically this is a very common way of showing the open chain form of sugars. So how you interpret a Fischer projection is that the substituents on either side, to the left and the right, come toward you, and the ones on the top and the bottom go away from you. And there's no atom that's in the plane of the screen, as it were. So this structure can be rewritten to look like this, so that the chain kind of bends over. And what we see is that the hydroxy group on the number 5 carbon can interact in a very facile way with the aldehyde that's at carbon 1. And when this reaction happens, it also gives hemiacetal, which is the pyranose, or the closed ring formation of the sugar. So even though there's lots of hydroxyl groups here, it primarily interacts with the one that's on the number 5 carbon. Does anybody know why that might be? The question is, okay, so basically the hydroxy group is going to interact with the number 1 carbon, which is the aldehyde. But there's lots of other hydroxyl groups here too. Why is it the number 5 one preferred? The 6-membered ring, yeah, that's right, because 6-membered rings are most favorable. They have the least amount of ring strain. So almost always, for this type of hexose, you will form a 6-membered ring. Now, depending on where the hydroxy group attacks the aldehyde, either from above or from below, remember this aldehyde, it's kind of drawn a little bit different to show you, to make room for the hydroxyl group here. But in actuality, an aldehyde is very planar, right? So if you think about if the aldehyde being in the plane of my hand, the alcohol can attack from either the top or the bottom. And so you get two different products. There's one in which the hydroxy group goes down, and there's one in which the hydroxy group goes up. And so you get two different forms. You get what's known as the alpha, let's say alpha D-glucopyranose and beta D-glucopyranose. Okay, so the number 1 carbon is thus referred to as the anomeric carbon, because you can get two different products here. This is also, so there's several stereocenters here. So this is what's known as the reducing end, because it's also reversible, but not so much as this one, because of the presence of the other groups that are decorating the ring. Now, these are called Haworth projections. They look a little bit closer to the actual 3D conformation, but they're not quite there. Okay, so The number 1 carbon is called the anomeric carbon, and so these two molecules, this pair, collectively are called anomers. So, okay, there's also a term that's used called epimer. Does anybody know what an epimer is? So if you have several chiral centers in a molecule, yes? If you change one of the stereo centers to a different configuration, then you get an epimer. That's correct. So these are also epimers, because the only difference between this structure and this structure is the configuration at the anomeric carbon. Okay, so I apologize if this slide's a little bit hard to read, but the central ring structure in hexoses is derived from pyran, and so they're called pyranoses. And this ring is a 6-membered ring with 1 oxygen. And so this, just like cyclohexane, this ring can exist in several different conformations. And there is a systematic way of specifying these, and they give them different names. And this is based on the Kramer-Pople ring puckering indices theta and phi. So what we see, so if theta, I apologize for this as well. I was going to change this on the slide. It says Q here, but it really means theta. So if theta is 0, or theta is 180, then you get what's called the chair forms. Everyone knows what a chair is, correct? So this is the back of the chair, this is the seat of the chair, and this is kind of the leg rest. And so these are numbered such that if you think about 4 of the central atoms being in the plane, one of the atoms will be above the plane and one will be below the plane. So for example, in this configuration, this conformation, the number 4 carbon is above the plane, the number 1 is below. And the way we write that is that the one that's above the plane gets superscripted and is written to the left, and the one that's below the plane is subscripted and gets written to the right. So this is a little bit hard to see, but I've written it down here. So this is the 4C1 conformation. So you would write it like this. And this one at the bottom, where theta is 180, this is the 1C4, because in this case, the number 1 carbon is above and the number 4 carbon is below. So when Q is 90, then you can get a bunch of different conformations. These are all different representative conformations. And you see that they're all labeled with either B or S. Anybody know what that might mean? So you have chairs, and then you also have boats, right? Have you heard of a boat? A boat happens when you flip one of the ring corners up or down so that you get both of them on the same side of the plane, if you think about the other 4 defining the plane. So this one is a boat, this one's 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 a boat. And S is skew boat. Yeah, exactly. So this is kind of like a bent boat. So, and these are also representative conformations, some of which are low energy and important for GAGs, particularly for idiuronic acid, where the 2SO conformation is important. But the nomenclature is very similar here. So for the boats, there will be, both substituents will be either above or below the plane. So both of them are above in this case. So it's the oxygen atom and the number 3 carbon. So this is O3b. Then, for example, let's take this one. This one would be 2SO, because the 2 carbon is above the plane, the oxygen is below, and the number 1, 3, 4, and 5 atoms are in the plane. Okay, so everyone Cool with that? Okay. All right, well, different sugars have different preferences for their ring conformation. So I've given a couple of examples here of glucose and iduronic acid, which are important for heparan sulfate and heparin. So, for example, we have alpha-D glucose. It can exist primarily in one of two chair forms. The one on the left would be 4C1. This is the number 1 carbon, 2, 3, 4, 5, 6. So this is 4C1. What happens is the chairs can interconvert. So if you flip this corner down and you flip this one up, you get the other chair, which is 1C4. Now what happens when you do that? Okay, so let's take a step back and talk about the substituents that are around the ring. So the number 1 carbon has a hydroxyl group, so does the number 2, number 3, number 4. Number 5 has this CH2OH group, as the number 6 carbon and a hydroxyl group. So if you think about ring conformation and cyclohexane in particular, the substituents that stick up above the plane and below the plane, does anybody know what those are called? They're axial, right? These are the axial positions. And then the other ones that point sort of away from the ring are which ones? Equatorial. Okay, very good. So for alpha-D glucose, where it's alpha because the hydroxy group is in the down position. So for alpha-D glucose, we have one of the hydroxyl groups in the axial position, and the other substituents are all equatorial. That's generally favorable. If a substituent has to have a choice of being either in the axial or equatorial position, which one do you think it would choose? Equatorial. Why is that? Okay, there's less steric hindrance. That's the answer I'm getting. So everybody must have remembered Dr. Desai's talk from last time. Very good. Or you had a good teacher in chemistry. Okay, so what happens when you interconvert chair forms is that all the equatorial substituents become axial, and vice versa. All the axial ones become equatorial. So in the 4C1 form of alpha-D glucose, there's only one hydroxy group, which is more bulky than the hydrogen substituents. There's only one that's in the axial position. All the rest are equatorial. If you flip the ring, then all of a sudden, there's 3 substituents that are axial and only one that's equatorial. So the preference is very much for the 4C1 form, as indicated by this heavier, darker arrow pointing to the 4C1. That's for alpha-D glucose. Now let's take a look at the alpha-L iduronic acid, or iduronate. Okay, so here we have the 1C4 form. Here is the 4C1 form. And here is another form that's also quite stable. It's called 2SO. This is just one of the skew boats. So let's take a look at the axial and equatorial positions. So in the 1C4 form, we have 1, 2, 3, 4 sort of bulky substituents that are axial, and only the carboxylate group at the 5 position is equatorial. In the 4C1 form, of course, everything is just reversed. And so now we have only the carboxylate group that is in the axial position, and all the others are equatorial. Now in the 2SO conformation, what happens is that essentially most all of the substituents are in what would be called the pseudo-equatorial form. So this minimizes the steric interaction among the substituents. So the 2SO form is actually very much preferred in idiuronic acid. And so that's important to know if you want to model GAGs that contain idiuronate, like heparan sulfate. Also, the 1C4 conformation is fairly prevalent. The 4C1 is not so much. And also there's OS2, which has a fairly low prevalence. Okay, that was monosaccharides. Now, of course, we know that GAGs are made up of disaccharide units. So here I show some representative disaccharide units for each of the main types of GAGs. So we have hyaluronic acid, we have chondroitin, dermatan sulfate, keratan sulfate, and heparin. So each of these contains a uronic acid. Okay, so it's in hyaluronic acid we have glucuronic acid, also glucuronic acid in chondroitin sulfate, and dermatan sulfate has idiuronic acid. Heparin can have either glucuronic or idiuronic. Just the glucuronic is shown here. And actually, in keratan does not have a uronic acid position. It has just galactose. The second monosaccharide is an amine, either glucosamine or galactosamine. And you can see those here. So there's a whole lot of similarity among these. And they are all acidic to some point. Hyaluronic acid is not sulfated, so it's the least acidic. But it does have a carboxylate group, which makes it acidic. So as Dr. Desai mentioned, these acidic groups are very important when you go to model these things, because they have very special characteristics which are not typical of many other molecules that you may want to model. You know, like if you think of a typical drug molecule, it may have an acidic functional group 1 or 2, but nothing to this extent. I mean, these are really, really acidic molecules. So we have to think about things, as Dr. Desai mentioned, we have to think about hydration, because the sulfate group carries a fairly large hydration shell. And so we'll talk a little bit about entropy and the effect of the displacement of water a little bit later on. Okay, so between the monosaccharide units, there is a glycosidic linkage. So this is, we talked a little bit about this the last lecture. This is the reducing end with the number 1 carbon. And I've just shown here 2 general monosaccharides. These are not GAGs necessarily. There's no uronic acid here or anything. But this just highlights the fact that the glycosidic linkage is made up of 2 rotatable bonds, and these are designated phi and psi. Now, as it turns out, because of the substitution pattern of the hydroxyl groups and the acidic groups and such on the sugars, hydrogen bonds can form in between the 2 monomeric units, and also the steric hindrance will cause the glycosidic linkage to adopt a fairly regular structure for at least some of the GAGs. So dermatan sulfate has a fairly regular structure. Heparan sulfate and heparin have a fairly regular structure because of these factors. And these give rise to helical structures in the extended linear structure. So because of this sort of consistent and regular value for phi and psi, we can use this as a constraint to limit the number of rotatable bonds during the docking procedure, as we'll see a little bit later. And here's a slide that I just show of dermatan sulfate and its 3 major types of helical forms, so that you can see that just like in amino acids, if you have a regular backbone structure, you get a regular secondary structure. For example, if the phi and psi angles of a protein or a peptide backbone are fairly regular, you might get an alpha helix, or you might get a beta strand. Well, in the world of sugars, usually you will get these helices that form. So here, this is an example. Dermatan sulfate has 3 major ones.
I have a question.
Yeah, sure.
If you go to the previous slide.
Yes.
What does that 213283?
Yes, the 2-1. This refers to the periodicity. The first number refers to the periodicity. And the second number refers to sort of the length of the repeat. So if you look at this one, you can see that, okay, this one is a side view, and this one is looking straight down the long axis. So you can see that it's perfectly a 2-1 helix is perfectly regular, and it has 2 symmetry, a rotation about the axis. So the 3-2 helix has periodicity of 3, and the 8 has 8. And the subscripts essentially refer to the length of the repeat unit.
So these numbers like 2, 3, 8, and these numbers come from the symmetry group symmetry, group theory.
It's two disaccharides per turn. So you finish a turn with 2 disaccharides. So it's like a tetrasaccharide will complete a turn.
So what is 3, 2?
3 disaccharides per turn.
Yeah. Yes.
3 disaccharides will finish at 360 degrees.
What about 2?
2 is 2 disaccharides.
No, I mean like 3, 2. What does 2 do?
I think the 2 is 2 turns. I think 3 disaccharides and 2 turns.
Okay. Yeah. Yes, that's my understanding. Yes. Okay. So finally, let's switch over to the protein now and talk a little bit about the binding site that's usually associated with GAGs. Now, remember that GAGs are very highly charged, negatively charged molecules. They're highly anionic. So you might guess that the complementary protein will have a high positive charge. So there are a couple of amino acid residues, arginine and lysine, which are typically charged, have a strong positive charge. And so typically you find arginines and lysines in GAG binding sites. So that's what I've shown here. This is a picture of thrombin, and it's anion binding exosite 2. The coloring, this is just a regular surface that I drew around the thrombin molecule, and the colors refer to the electrostatic charge. So sort of the blue and purple regions refer to the positively charged regions, and then the orange and red ones are the negatively charged regions. So you can see that there's a fairly extended group of basic residues here which will impart a very high positive charge to the binding site. Also, GAG binding sites tend to be fairly surface exposed and shallow. They're unlike many other binding sites for drug molecules that are kind of deep pockets. So unfortunately, my other computer isn't working, or I'd have a kind of an animation to show you that this site is really quite shallow. You can have some grooves and things in there like that, but generally they're fairly shallow. Okay, so now we know something about the protein and the ligand. So what drives the interaction between the two? Well, you can all take it all back to thermodynamics. So this basically is the driving force for everything in the universe, including interactions between macromolecules and ligands. And basically, what happens is the receptor, the affinity of the ligand for the receptor is determined by the free energy of binding, which is delta G. And that is that can be written delta G equals delta H minus T delta S, where delta H is the enthalpy, change in enthalpy, and delta S is the change in entropy, which is also equal to minus RT ln of K, the equilibrium constant. Equilibrium constant tells you how much of the product you have, which in this case is the complex ligand, receptor-ligand complex, versus the individual disassociated components. So let's talk a little bit about each of the terms individually. So enthalpy refers to the change in enthalpy is determined by the formation and breakage of inter and intramolecular chemical bonds. So for the most part, we only need to be concerned about the weak non-covalent interactions, which we can think of as kind of bonds in quotes. However, there are exceptions where covalent bonds are formed. There's suicide inhibitors and affinity labels and such. And also, in the world of serine proteases, such as thrombin and antithrombin, for example, there's an association that takes place. There's a reaction that takes place. And that's basically how the inhibition takes place. So let's talk a little bit about the weak interactions that take place. So we have several different kinds. They're all basically dependent on charge and the distribution of charge of one interacting species versus the other. So if we have two atoms that are interacting and one has a positive charge, one has a negative charge, we can have an attractive force that is called an ion-ion, or simply an ionic interaction. Now, you can also have a situation where, due to differences in electronegativity of the individual atoms, you can have what's called a dipole set up, where there is a partially negative charge on one of the atoms, and the other atom has a partial positive charge. And in the world of electrostatics, opposites attract, so you can also have an interaction between an ion and a dipole. For example, in this case, the positive ion is interacting with the partially negatively charged oxygen atom of, let's say, a carbonyl oxygen. And also, dipoles can interact with one another by placing their partially positive and partially negative charged atoms in a complementary fashion so that the opposites attract. Now, a hydrogen bond is kind of a special case of a dipolar interaction. What happens in a hydrogen bond is that you have one atom, which is called the donor, the hydrogen atom itself, and then one atom, which is called the acceptor. Now, the donor and the acceptor atoms are usually heteroatoms that have a higher electronegativity than the intervening hydrogen atom. And so what happens is, in the donor, there is a dipole that's set up wherein the proton or the hydrogen has a partial positive charge and the donor has a partial negative charge. Well, if it comes into contact with another atom that also has a partial negative charge, it can act as what we call a hydrogen bond acceptor. And there can be a beneficial, sort of attractive interaction between the two. And then we have what we might call generally dispersion forces. You might also heard this called pi stacking, where essentially you have a molecule or a part of a molecule that can be induced to have a dipole. In other words, it has some cloud of electrons that can be, wherein the electrons can be pushed or pulled in order to set up a dipole within the electron cloud. And so, for example, one side, let's say one side of an aromatic ring, let's say, could have a partial positive charge, and an adjacent one that could have a partial negative charge. They could beneficially interact with one another. So, and another kind of special case of this is the pi-cation interaction, where a positive ion will interact with the negative charge that's associated with the electrons in the cloud. I also include halogen bond here. This is generally not so important for GAGs, so I won't go into it now, but it's another case of sort of a dipole-dipole interaction. So essentially, the bonds, quote, that are formed when the ligand binds to the receptor will stabilize and thus favor the ligand-receptor complex. And the bonds that are broken when the ligand binds will disfavor the formation of the complex. Okay, then the other component of the binding free energy is the entropy. And entropy refers to the amount of uncertainty in the degrees of freedom in a closed thermodynamic system. That means how easy is it for molecules to move around. It's the degrees of freedom. So in terms of biological molecules, there are two main factors that account for this. First of all, we have the loss of translational and rotational degrees of freedom, which means the position of the ligand, as well as any freely rotating single bonds within the ligand, are more or less fixed in place when it binds to the receptor. They can't really move around. So they've lost that degree of freedom when they bind. So this results in a decrease in entropy that will disfavor the formation of the receptor-ligand complex. Then the other aspect is what's known as the hydrophobic effect, which you can really think of as being a special case of the first one, in which organized water molecules that surround the hydrophobic portions of the ligand and possibly inside the binding site, actually very often that's the case in more buried cavities, that are displaced during the docking event result in an increase in entropy. The molecules are freed up and now they're able to move around. So this is an increase in entropy that favors the formation of the receptor-ligand complex. So I show here a simple kind of schematic figure with a binding site with some water molecules that are being held in the binding site. Here's the ligand, which also is hydrophobic portions are surrounded by what's known as a clathrate structure of water molecules. When the two associate, there's a release, there's an increase in entropy because the water molecules that were being held in place are all of a sudden freed up. And so this will favor the binding. Yes?
So that ligand is hydrophobic on the surface, right?
Yes, this is a theoretical hydrophobic ligand, yes.
And how come then water will surround? Because water is very hydrophilic.
Exactly, because notice that the water, it doesn't want to interact with the ligand itself. Right. Because it has nothing to grab onto. Water is very polar. Lipophilic surfaces are very nonpolar. So the polar water molecule doesn't have anything to hydrogen bond to or grab onto. Instead, what happens is it hydrogen bonds with itself. And so what happens is you get sort of an organized sort of water cage structure that surrounds the hydrophobic portions. This is called the clathrate cage. And that's what I'm showing here. That gets disrupted when the ligand binds. Okay, so now how do we translate all these ideas into something practically useful that we can use for modeling? Well, one of the main ways, and it's used extensively in molecular modeling, is molecular mechanics. So the idea here is to treat molecules as physical entities that are governed by the conventional, that means non-quantum or non-molecular orbital Newtonian laws of physics. And so what you see here is a rather long and imposing looking equation, which is actually really simple when you think about it. It's just a summation of terms that correspond to the different energy terms or forces within the molecule. And so the total internal energy of a molecule is the sum of simply various energy terms representing various stresses and strains. So let's take a look at these individually. So actually, let me just kind of briefly mention what these are. We have a stretching term, which means that two atoms that are bonded together will they're dynamic, they're moving, right? So they're kind of vibrating and they're stretching. And there's forces that are associated with that. So there's a stretch one. If you have three atoms, you have a bend that can happen. If you have 4 atoms, there can be torsions and out-of-plane interactions. And there's also van der Waals and Coulombic interactions. So let's talk about the stretch first. So here we have 2 atoms that are bonded to one another. So how do you think what do you think a good way of representing the structure would be if you think about a molecule that's kind of vibrating? I just show kind of a stick here, but that's obviously not going to do it because we know that the molecules aren't completely rigid, except at absolute 0. In which case they'd probably just fall apart. So what would be a good way of representing sort of the force between these 2 atoms? A spring, very good. So let's put a spring in. Okay. So we can put a spring in between the 2, and we can measure the distance between the 2. And what happens is that the spring has an ideal length that it wants to be. This is the ideal bond length. So if you try and pull the atoms apart, the spring will try and pull the atoms back together. At the same time, if you try and push the atoms together, the spring is going to want to push them back apart. So there's an ideal distance whereby if the distance decreases, the energy goes up. And if you increase this distance, energy also goes up. So there's an ideal value here that is the ideal bond length between any 2 atoms. And it varies depending on what the atom type is. So this is just given by this equation here. Basically, if you want to calculate what the stretching term is for a molecule, you basically look at all the bonds in the molecule, and you look at what their distances are, and you square that term, and then divide by 2. So this gives you the stretching term. You might also know, have you heard of the harmonic oscillator? Yeah, so this is what this is. And the equation is 1/2 kx squared. That's what this equation basically is. Here's the 1/2 k and then x squared. Here's our x. It's r in this case. And this is known as Hooke's law. Hooke was a British physicist who lived in the 1600s. He came up with this. Okay, so that's if you have 2 atoms. What happens if you have 3 atoms that are bonded together? Well, now, in addition, you can have a bend term. This would be basically like this. So, again, we could think about this in terms of sines and cosines and defining it in a very complex way. But there's a simpler way to do it, which, of course, is just to use the spring again. And we can see that the form of this equation is pretty much exactly like the first one, where there's going to be an ideal bend angle. If you try and squeeze them together, it's going to resist. If you pull them apart, there's going to be also a force, which is going to want to restore the ideal angle. Okay, and then if we have 4 atoms, we can have a couple of different things. First, we might have a torsion angle. Now, a torsion means twisting. So if you have 4 atoms connected together, if you try and turn this bond, there might be some resistance to that. And this is called the torsion angle. And unfortunately, we can't use a spring in this case. We have to resort to sines and cosines. And you don't have to necessarily worry about what all these mean. But depending on what substituents are on these 4 atoms, you can have different forms for different periodicities, we say, for how easy it is to rotate that central bond. And then finally, we have OOP, not OOPS. We have OOP. OOP is out of plane bending. So if you have 4 atoms that are connected such that one is in the middle and there's 3 others on the edge, there might be a case, like let's think about our aldehyde, where it wants to be planar. And if you try and make it non-planar, there's going to be some resistance to that because of the hybridization of the orbitals, and it's going to want to be planar. So again, we can use the spring to define a distance d, and it works just as exactly the same as the other one. It's like a harmonic oscillator. But there's a restoring force that is going to want to keep the 4 atoms all coplanar. Let's see, am I doing on time? Okay, still bad. Okay, then in addition to all these bending and stretching terms, there is what we call van der Waals interaction. Now, van der Waals interactions can occur between any 2 atom types. And it occurs because the electron cloud that surrounds the nucleus can be polarized. In other words, the electrons don't always have to be uniformly distributed around the nucleus. There can be more on one side than the other. And these electrons can be attracted by, let's say, a positively charged atom that comes in close proximity. So what we see here is that if 2 atoms are sufficiently far apart, they don't really see each other, and so nothing happens. But as they come closer and closer, you start to feel the positive charge of the other one, in which case some of the electrons in the cloud will be pulled toward it. And if you bring them even closer together, the nuclei will start to feel each other, and they'll want to push each other apart. But at some point, there will be an ideal distance between the 2 where the dipolar coupling is ideal. And this is called the van der Waals radius. And there is an equation that's commonly associated with this force. It's called the Leonard-Jones equation, or the 612 equation, because it is dependent on the 6th and the 12th power of the distance between the 2 atoms. So the 12th, the distance to the 12th term is the repulsive force, and the 6th is the attractive force. And so if you think about this, here's 2 neon atoms. They're really inert gases, but even inert gas atoms can be attracted to one another by virtue of this van der Waals force, because the electron cloud can be polarized. So we see as they come together, sort of the lack of electrons on one side will induce a positive charge, and in the neighboring atom, the electrons will be attracted to this positive charge, and so you have an attractive interaction taking place. Now, if you were to make a graph of this equation, you'd see something like the following. So at this point, this is the ideal distance between the 2. This is, in fact, the van der Waals radius. If you decrease the distance, they're going to really want to push each other apart. And it doesn't take too much distance for them to start really feeling each other, and so the energy shoots way up. As you pull the atoms apart, the attractive force becomes less and less and less until after you get to beyond a certain point, they don't really feel each other. And then the final term that we commonly use in molecular mechanics is the Coulombic force. And Coulombic forces arise purely because of interaction of charges. And here, as always, the law is opposites attract. So if one atom has positive charge and the other has a negative charge, they will have an attractive force. If they're both positively charged or they're both negatively charged, there'll be repulsive force. You can call back later. Sorry, I'm busy. Okay, so opposites attract. Now, the strength of that interaction will be dependent on a couple of things. It will be dependent on the radius, this distance r, the distance between them, and which means that the closer they are together, the stronger the force will be. And as you pull them apart, the interaction will be less and less and less because they'll feel each other a little bit less. The other thing that can happen is that, well, as it's shown here, it's kind of just empty space. There's nothing in between them. Most often, what happens in biomolecular interactions in particular, there will be lots of, let's say, solvent around, let's say water, that could come in between the two interacting. Okay. They can come in between the two interacting species. This is called the dielectric. And substances that have a high dielectric constant will be very good at shielding the two species from one another. And these have an insulating effect. So water is one of those substances with a very high dielectric constant. It's very polar. And so because it's polar, it will be able to interact with the individual charges very well and shield them. And again, these are called Coulombic forces after the French physicist de Coulomb. So where do the charges come from? First of all, we have to start with electronegativity. So as you all remember from general chemistry, hopefully, the electronegativity is the ability or how much an atom actually wants an electron. And you might remember that the elements to the right and to the up and to the top of the periodic table have high electronegativity. And the ones that are to the left and lower have lower electronegativity. And there's reasons for that, which we really won't go into now. But the practical upshot is that we can have what's called polarizability happening. So that if you have two atoms that have different electronegativities, again, if they're far apart, they don't really feel each other. As they come close to one another, there is a dipole that's induced because the more electronegative one wants to pull more of the electrons toward it. And so it ends up having a more negative charge. And the one that is less electronegative gives up its electrons, and so it becomes positively charged. Okay, so what that means is that each atom in a molecule can have a slightly different partial atomic charge. So, for example, in benzene, all of the carbons and all of the hydrogens have the same charge because they are equivalent. However, if you put something like methyl ester on, which is an electron withdrawing group, we see that because they are connected to one another, the oxygen atoms have a greater electronegativity than the carbon atoms in general. It will pull electron density away from the carbon atoms in the ring, so that it will have a relatively smaller or more negative partial charge. In a similar fashion, if you put something like an NH2 group on here, which is electron donor, you will increase the negative charge of the remaining carbons in the ring. Okay, so now we basically have the basic equations. And now how can we bring a computer in to actually do the calculations and have something useful for us? Okay. We can create what's called a force field. So together, the equations that I just talked about create what's called a force field that describes how to calculate the potential energy of a molecule. And the bond lengths, bond angles, and atomic radii are taken from high-resolution X-ray crystal structures of small molecules and biomacromolecules. And the force constants, which are the Ks in those equations, are taken from infrared spectroscopy and similar experiments. Okay, so then we have to talk about energy minimization. So energy minimization is essentially the process of structure optimization. can be thought of as a simultaneous process, because when you minimize the internal energy of a compound, you make it more stable, and it has a more favorable conformation. So by definition, the most optimal structure is the one that's at lowest energy. Of course, this means in vacuum at 0 degrees K, which is not very realistic. So although there are a variety of permutations on the algorithms, mostly to increase computational speed, the basic idea is if the energy is decreasing, then the structure is improving in terms of its stability and preferred conformation. But it's not quite so simple, because there might be several different minima that you might have. So let's think about cyclohexane. When you draw cyclohexane into the computer, you just draw 6 atoms, and they're all in the same plane. So it's a planar structure. And that's a relatively high energy structure, because the tetrahedral atoms don't want to be all coplanar. Instead, they would like to form a lower energy structure. So when you do energy minimization, the program will change the bond lengths and angles so that you find an energy minimum. However, as we know, for cyclohexane, there are multiple low energy conformations. So you might start at a high energy conformation, such as what you might sketch into a modeling program. You might tell it, OK, do the minimization, and and it does it. OK? And you will reach a minimum. But it might not be what we call the global minimum. It might not be the most preferred or most energetically favored structure, because it's kind of the law in computational chemistry is that an energy minimizer won't go up an energy hill. So once you reach this bottom part, you're basically stuck. So if you reach you might reach what's called a local minimum, which is a low energy structure, but it's not necessarily the lowest. So that would be representative of the boat conformation. And so if you were to have drawn your molecule and its structure started somewhere over here and you did the energy minimization, well, then you would get the global minimum, which would be the chair form. But you wouldn't necessarily know that you have the lowest energy structure a priori for more complex molecules. So how do we get around that? Well, you can do a couple of different things. You can do an exhaustive search where you define the structures of each of the different conformations and calculate the energies for, quote, all of the conformations. And then you just compare them, and the structure with the lowest energy is the global minimum. And this process generally involves varying the torsion angles for the, quote, rotatable bonds. Rotatable bond is one that you can twist and change the conformation of the molecule without a high degree of difficulty. So it's relatively easy to move a rotatable bond. So generally, single bonds are rotatable. Double bonds are not rotatable because they want to be coplanar. So as an example, at a 15-degree increment, the number of structures would be 24 to the n, where n is the number of these bonds. So it shouldn't be too difficult to see that if you have more than just a few bonds, this number gets to be really exponentially huge. And so even the fastest supercomputers won't be able to tell you what the global minimum is, even for a relatively simple molecule that's of moderate size. So another way to address this is used molecular dynamics. And simply this means adding heat, which means forces to the atoms in your simulation, and time to molecular mechanics in order to break out of the local minima. And this has to be used with larger molecules, of course, which have a lot of rotatable bonds. Okay, so let's talk about these rotatable bonds. So what I've shown here is a disaccharide unit that's a common component of heparan sulfate. So we have the idiuronic acid here in its 2SO conformation. And here we have the glucosamine, which is acetylated at the 2 position and sulfated at the 6 position. And this is in a 4C1 conformation. So as you can see, there's intramolecular hydrogen bonds that can be formed that kind of restrict the conformation of the molecule. So let's take a look at what bonds might be rotatable. Okay, so let's look at this one. Do you think this one is probably rotatable? Even though it's hydrogen bonded, it's possible that you might have a conformation in which these hydrogen bonds aren't there. So that one's probably rotatable. How about this one in the middle? How about these in the middle? Are those rotatable? Yeah, generally they are, but not so much. As I alluded to earlier, they tend to take specific values because of this sort of intramolecular hydrogen bonding and steric effects that take place. How about the hydroxyl group? Is that rotatable? Sure, that's really rotatable. But what is attached to the oxygen atom? It's just a hydrogen atom. So that's not very much. In general, when you do modeling or docking, that sort of thing, when you have just a hydroxyl group, you don't need to consider it explicitly as a hydrogen bond because wherever the molecule ends up bound to your protein, it will be able to easily find where the hydrogen should be. So you don't normally have to really consider that to be a rotatable bond. How about this one? This is certainly rotatable, right? It can move around. And this one, and this one. So you have potentially 3 more rotatable bonds here. This one can rotate. How about this one? This one right here. This is the amide linkage. Probably not so much, huh? Amides want to be planar. So this is not rotatable. Okay, how about this one? Yeah, that's rotatable, right? But again, all you have here is just nothing but hydrogen atoms. And they're not really polar or anything, so they don't necessarily form a directional interaction. So we don't normally think of terminal methyl groups as being rotatable. So basically, you can have anywhere from 7 to 15 rotatable bonds, depending on how you count them. And of course, this becomes an issue when docking larger oligosaccharides. Typically, we find that when you get to about hexasaccharide, those can be modeled fairly reliably, but if you go larger, octasaccharide and larger, then things kind of fall apart. If you try and, let's say, do a docking with those, it's really going to give you bad solutions because it just can't handle the conformational space, the number of rotatable bonds. However, we can use the rigid backbone approximation to partially address this, and this is what we've done previously. Okay, then of course we can use molecular dynamics, and this is a very popular way of treating molecular interactions where you want to keep the ligand completely flexible, also the protein to which it's bound. So this involves typically immersing the protein in a solvent, placing your ligand there, and then adding heat means the computer will move the atoms around according to the terms in the force field. And over time, you can actually see how the molecule interacts with its protein. I won't say too much more about that. I mean, we don't really have the time to talk about it. It's an important area. It's very useful, I think. But that's molecular dynamics. So the other sort of really important way that you can model the molecular interaction is by using docking. So essentially, the docking problem involves finding an optimal complementary fit between a ligand molecule and a suitable binding site and receptor. And this typically involves 3 separate operations, which may or may not be separate, but each has to be performed. So first of all, the program has to generate plausible ligand conformations. And so in general, the conformation that the ligand adopts when it's bound to the receptor is not known. So the docking program must therefore generate a number of possible conformations to test. So as we've just seen, this might be something kind of difficult to do if your molecule has a lot of rotatable bonds. So programs will use things like dictionaries, lookup tables of preferred torsion angles and things to assign plausible low energy conformations to the ligand. By the way, I just talked about finding the global minimum, finding the lowest absolute lowest energy conformation of a molecule. Do you think that a ligand has to be in its absolute lowest conformation in order to effectively interact with the receptor? No, it can probably be a little bit higher energy. But for large, complex molecules, there is a lot of low energy structures. It doesn't necessarily have to be the absolute lowest one, but it should be sufficiently low such that the molecule is in that conformation for a significant amount of time. Otherwise, it doesn't really work as a ligand. Okay, so the second thing that the docking program has to do is place the ligand into the binding site. So yeah, it must be able to place it in the binding site such that the distances and angles between the complementary functional groups, they're chemically meaningful, and that the atoms are not clashing with one another. And the third thing that it has to do is it has to be able to tell which conformations are good and which ones are bad. So it has to assign a score or fitness value to the docked conformation. So in ligand docking, it's treated computationally as a mathematical optimization problem. Thus, the docked solutions or poses that are generated must be evaluated by a fitness or scoring function to determine the quality of each pose. So there's lots of different scoring functions, but they tend to have sort of a lot of things in common. In other words, they'll have terms that account for the hydrogen bonding. They'll have terms that account for the van der Waals, the hydrophobicity, and even some that will try and include some terms for entropy. But in general, it's very, very difficult to predict the binding affinity a priori, just by looking at the docking score. So one of my colleagues mentioned that docking is easy, but scoring is hard. So when you do the docking, you don't want to focus exclusively on the score because it might mislead you. You should look at some other things like the consistency of the docked pose and how chemically meaningful it really is. Okay. So to take docking a step further, we can do what's called virtual screening, which is where you dock a huge library of molecules. And so that's what we need in order to encode or capture this phenomenal diversity that GAGs have. So for example, heparan sulfate has a uronic acid, which can either be glucuronic acid or idiuronic acid. If you have your idiuronic acid, it can be in one of 2 major conformations. The idiuronic acid could be either sulfated or not at the 2 position. The other part of heparan sulfate is the glucosamine. That is usually in the 4C1 conformation, but it can be sulfated at the 2, the 3, or the 6 position. So there's a lot of variability in here. And so what happens is that you end up forming a library of lots of compounds, tens and hundreds of thousands of compounds very, very easily. So we need a way to actually screen these very effectively in order to find out which ones might have good specific interactions with the target protein. So just as an example, if we have a hexasaccharide library of heparan sulfate, there are 13 naturally occurring idiuronic-containing disaccharides. You multiply that by 2 because idiuronic acid can be in 2 major conformations. So that's 26. Okay, and then there's an additional 10 naturally occurring glucosamine-containing disaccharides. So if you add the 10 to the 26, you get 36. That's the disaccharides. And then if you want to have a hexasaccharide library, that's 3 disaccharides. So it's 36 times 36 times 36 is almost 50,000 compounds, which is a lot, particularly since they're kind of large molecules. So virtual screening does provide a way to dock many GAG sequences to a given protein. It would select the ones that bind with high affinity and selectivity. However, we can't spend a lot of time focused on each and every one, because if we did that, it would take like months or years to get through a single library. So we have to have a kind of a two-stage strategy where we have 2 separate filters. The first one is kind of quick and dirty. It's done using, well, we use GOLD, and GOLD uses a genetic algorithm. And in a genetic algorithm, the greater the number of iterations, the longer time it has to find a good solution. But we can't have a high number of iterations early on, because then, of course, it would take forever. So we have to cut down on the number. So we use 10,000 iterations initially, to which we subject all of the compounds to. And then we select, based on the scoring function, we do select, for example, the top 1%. And if they're in the top 1%, then we keep them. If they're not, they get discarded. And the ones that we keep go on to a second filter, which we dock them much more rigorously. So it's 100,000 iterations, and we do not 1, but 3 runs per sequence. And we compare the top 2 solutions from each of the 3 runs in terms of their root mean squared distance. And that means how consistently are they docked. If they're all docked in exactly the same way, then the RMSD will be very low. So if it's below 2.5 angstroms, which is our cutoff value, then we keep them because they are consistently docked. And the consistently docked sequences imply that the interaction is specific. In other words, because they're docked consistently, there's some arrangement of the side chains in the protein that specifically recognize the particular sulfate groups, what carboxylates, whatever, on the GAG that it's interacting with. So this we use as a second specificity filter. And the ones that meet this criteria, we keep. Those are called high affinity and high specificity sequences, purportedly. So, and then the others, of course, are discarded. Okay, so this general scheme is very powerful. It allows us to theoretically provide us with the tools to model any GAG protein interaction. And as I mentioned, it's powerful and easy to do, but there are some caveats when using virtual screening with GAGs compared to normal molecules. First of all, I mentioned the rotatable bonds. You have a fairly large molecule with lots of different rotatable bonds. It can have lots of different conformations. That makes it really difficult compared to a smaller, like a drug-like molecule. And we also need to be cognizant that the side chains in the protein can also move around. And that's a limitation that many programs still have, but it's becoming increasingly common to be able, now that computer power is sufficiently high, we can start to model the flexibility of the side chains as well. Yes, Prem?
In your earlier slide, where does the flexibility kick in your algorithm? At which step do you start introducing the flexibility rather than rigid body dock?
Well, but when we dock, we keep all of the rotatable bonds free except the glycosidic linkages. Even from the first step, yes. Mm-hmm. And at this point, we keep the side chains in the protein rigid, because we know that this is a known conformation that might recognize a particular pattern of sulfate groups, for example, on a GAG. Another thing to be concerned, not worried, but cognizant of is that the ionization state of the charged groups. For example, if you have two acidic groups, which are normally deprotonated and charged, if they're next to one another, it might make sense that one of them is protonated so that it can form a hydrogen bond between the two. We don't really know that necessarily. And to be able to calculate the affinity for the receptor fairly well is probably something we'll need to look at. But it's an approximation that we make now is that all of the acidic groups are ionized. Also, the involvement of water is important. As Dr. Desai mentioned earlier, these anions, particularly sulfate groups, are associated with a large cluster of water that's associated with it. And displacement of that water, again, releases entropy, increases the entropy, and will aid in the binding of the GAG to the protein. So an interesting side story in that is that we did a study recently and published in PLOS 1 where we were analyzing the characteristics of the antithrombin and thrombin binding sites. And we know that antithrombin recognizes a very specific heparan sulfate sequence known as the pentasaccharide sequence, or DEFGH. And we know, for example, that the presence of particular sulfate at the 3 position of the F subunit is very important for the recognition. And what we found is that when we analyzed the binding site for the pentasaccharide in the antithrombin, in the antithrombin binding site, we see that there are several water molecules that are in this sort of groove here, some of which are highly conserved. They're tightly held. A couple of these are fairly tightly held. And so displacement of these by the GAG is going to release the water molecules, increase in entropy. It's going to affect the binding in a very positive way. So something very interesting that we found when we looked at the crystal structure and also when we did docking of other GAGs to this binding site, we saw that interestingly, it is the 3-position sulfate that displaces one of these water molecules. So that's kind of an interesting, kind of cool thing that we found that deals with the displacement of water and its involvement. Yes?
Yeah, so it has been mentioned that you get this bond length on energy and, atomic radius and bond angle. You get it from X-ray crystallography.
Yes, we take the measurements from high-resolution crystal structures.
If you don't have crystal, can you do modeling? Or
yes, sometimes that's done for some molecules that aren't represented well in the crystallographic databases, particularly those with kind of different atoms, like some of the metals and the transition elements. They're not really well represented. So those kinds of things can be treated fairly well with high-level sort of quantum mechanical calculations. And so the bond lengths can be calculated using that way. And then the results applied to a molecular mechanics force field, let's say. Yeah. Okay, so I think it's just about getting close to lunchtime, and I'm sure everybody's kind of ready for lunch. So I'll just mention, before I close, one case study that we did. This is one that we did initially to sort of, it's a proof of concept of our virtual screening algorithm, where we had a library of heparan sulfate okay, so a little bit of background. So as I mentioned before, the pentasaccharide sequence of heparan sulfate, that is the DEFGH sequence, binds to antithrombin with high specificity. So we generated a library of nearly 7,000 hexasaccharide sequences that included DEFGH and also similar highly sulfated molecules. Generated a virtual library of those and screened them using our two-stage filter algorithm, in which antithrombin affinity was the first filter and the self-consistency of docking was the second, as I mentioned before. And only 10 sequences got through to the final, got through both filters. And we found that the results do explain the biochemical observations with a single substitution and ring truncation variants of the pentasaccharide. So here's our 10 solutions. We see that, okay, our docked solutions really well match the X-ray crystal structure. Okay, here is, again, an overlap of our 10 sequences. There's 9 that essentially match the crystal structure, and there's 1 that was kind of backwards, indicating that we might have a novel pharmacophore, something where we could build a molecule, a slightly different scaffold with slightly different positioning of sulfate groups. But we can see that the central DEF sequence was retained in all of the molecules that made it through both filters. And then we also were able to correlate a version of the gold score with the actual measured binding affinities for some DEFGH variants to antithrombin, native antithrombin, and a couple of antithrombin variants. Okay, so just to kind of conclude, I hope I've shown that molecular modeling techniques can provide a powerful and general method to address GAG protein binding, and that these tools can be used to identify potential high affinity, high specificity GAG sequences for any potential GAG binding site. Okay, so thank you for your attention. I'd like to acknowledge a couple of people. Of course, Dr. Ramesh Desai, who is the program project leader, and who's given me a lot of guidance and also a few slides in my presentation. Also, Dr. Glenn Kellogg, who is our advisor for this project, and also computational chemist in our department, also gave me a lot of support and also some slides for this presentation. And of course, to all of you for coming and listening to my talk. And hopefully everybody's excited to do some modeling this afternoon and try out some of this stuff yourself. And of course, I can't ignore our grant here, which I think is not the right one, but actually, so this is not this is not the NIDA grant. This is of course our program project from the Heart, Lung, and Blood Institute. So once again, thank you all for coming, and I think I have 6 minutes left if anyone has a question. Yes?
You said that you talked about rotatable bonds and rotatable bonds at room temperature, because you know that temperature increases.
Exactly. So that's one of the things that you can do in molecular dynamics, is that you will specify a temperature. And normally, when people model biological systems, they want to model it at either room temperature or more commonly physiological temperature. And the higher the temperature, the more energy you add to the system, and the more the bonds will move and shake around. And so the more they move around, the better the chance they are of jumping out of one of those local minima. So the idea is to run the molecular dynamics simulation for some time, and then at certain points you can see, as the molecule interacts with its protein, all of a sudden you monitor the energy, and all of a sudden the energy will kind of drop because it will have found a better conformation. And so at that point you can take a snapshot and then energy minimize the whole system, and you will get a representation of what hopefully isn't the actual interaction taking place between the protein and the ligand.
And also you have discussed about all the single bonds. There's no double bond in all except like carboxylates.
Well, there are double bonds, of course. Then GAGs have acetyl groups, and there's things like that. So, and also, bonds that are part of a ring aren't really considered to be rotatable, because they're not really freely rotatable. They can rotate, but since they're in a ring, they all have to sort of move. All the bonds in the ring have to move in a concerted fashion. So,
like a hypothetical question. Take like cis-trans isomerization of retinal, for example.
okay.
And with its interaction with protein,
Mm-hmm.
like cis-trans retinal. So can this molecular modeling do that cis-trans isomerization?
Well, typically, now that's a good question, very good question. Typically, you can't model things like chemical reactions with simple molecular mechanics. Whenever there's sort of a change in the bonding structure, you have to go to quantum mechanical methods. And the cis-trans retinol case would be an example of that, where the retinal has an extended conjugated pi system which is able to absorb a photon, and then you have some high energy intermediate which does have a rotatable bond that will affect the isomerization. But typically that type of thing has to be treated with quantum mechanical calculations, which are computationally quite a bit more expensive. Yeah. Karthik.
When you're rotating the bonds around to see if you can find a lower energy state, why not do something even more random? Why not do like 10 bonds at the same time? Or do you guys do that?
Well, I mean, that's part of the way that these docking programs, for example, work, is that, well, gold, which you'll work with tomorrow, actually uses a genetic algorithm to generate different conformations. So what happens there is the program will generate a population of just random conformations. And then it will score each of those conformations. And then the ones that get scored highest, they get kept. And sort of those are considered to be the parents. And the values of those rotatable bonds are mixed and matched for those highest scoring ones to generate what are called children conformations. And then they are evaluated, and the top ones are kept from that population. And so the cycle continues and continues. That's what I mean about genetic algorithm iterations, is it will go for 10,000, or it'll just try and improve it over 10,000 or 100,000 iterations. And then at the end, hopefully you have some very favorable conformations, which interact well with the protein, for example.
So you have made it more efficient by using...
Oh, exactly. And there's no way that you could do it otherwise. You have to be smart about it, basically. Yeah. Yeah, Spencer.
You said the glycosidic linkages are typically considered fixed?
Well, sometimes they are. That's not generally the case in general for all GAGs. Probably it's not so much the case with, for example, hyaluronan, but for the more highly substituted ones, that have bulky groups that can also engage in hydrogen bonding, such as like heparan sulfate or dermatan sulfate. There's restrictions on its movement. And we know from NMR experiments and such that the values change relatively little.
Do those like bond limitations ever cause problems in the case of, say, a model of induced fit when the linkage is binding by induced fit?
Well, yeah, that is an issue. I mean, that's what we model is typically what might be considered an initial interaction. Doesn't necessarily take into account induced fit. In order to model something like that, you would have to be a little more sophisticated. You might try using molecular dynamics, although if the process is inherently slow, you might run the dynamic simulation for a long, long time and not really see anything happen. So yes, that's also kind of a limitation. So that whenever we do these docking exercises, it's always sort of, the complex that you get is always considered to be sort of an initial recognition complex.
I actually want
Yeah.
to comment on this. So even if we consider it a conformational selection, not induced fit model, when we start by one protein model that we start the docking with, this protein model is the one that was crystally taken from a crystal structure. And it is with an octosaccharide ligand, the one that we're working with.
Right.
Certainly, if you're modeling induced fit, you would actually have to have structural information on both conformations, because the computation, unfortunately, is not strong enough to transition from one form of the protein to another unless the degree of induced fit is really small.
Or you have some other large amount of biochemical data which can sort of guide you into as to how the protein actually moves.
So how well you can mimic the nature environment, and then how accurate this compared with the experimental?
Well, it can be done fairly accurately, and there's a lot of cases where, for example, molecular dynamic simulations and dockings have
in nature also we can count like a lot of salt, something like that. So this is like calcium salt, like do they affect the binding?
salts.
Yeah, for example,
Yep.
calcium and potassium salt.
Yes, well, those calcium ions can affect the binding in a couple of different ways. First, if it's in fairly high concentration, it might act as a buffer between the ligand and the protein. But also many proteins have metal binding sites. Like thrombin, for example, has one for sodium. There's a sodium effect where if there's sodium present, there's what's called an allosteric effect. And we'll hear a lot more about allosterism tomorrow, because Dr. Desai will talk about allosterism. But yes, the binding of a metal into a protein can change its conformation significantly. And so we have an allosteric effect. That can be modeled again if you have enough knowledge about the protein itself to be able to model the different conformations. Okay?